Okay, well, thank you everyone for joining us. This is the last IMAG MSM working group on multiscale modeling for viral pandemics of April. It's hard to believe that it's already turning into May. We have two great speakers lined up today. They both very generously agreed to stay for discussions afterwards, and depending on the tenor of the discussions, we'll either use the breakout rooms or have an open pit to discuss. As usual, I need to remind you that this content is being live streamed on YouTube. It's being recorded, and these recordings will be archived and made publicly available. The usual preamble that we have myself, James Glazier, and Reinhard Labenbacher as co-leads. Reinhard is driving today, so he's not able to attend the meeting. He sends his regrets. We have Jim Sluka and Bruce Shapiro on the call. You've had interactions with them, and I hope they'll help you in any ways that you need. If you have any issues, concerns about anything to do with the meetings, please do let us know, and we'll do our best to address them. We have the various modern means of communication. We definitely appreciate your help in publicizing our work. And we have a Slack channel, Twitter channel. We have the official IMAG MSM wiki pages, which always need love and attention. There's now an IMAG LinkedIn page to which you can refer. Please consider joining that. And we have the YouTube channel, which distributes our videos. So please help us out. We have great talks. We have great speakers. Please help us reach the broadest audience possible for this wonderful work. And now, following our recent protocol, I'd like to open the meeting for any quick announcements. I have an announcement, which is that we'll have our regular steering committee meeting tomorrow at 11 a.m. If you think that you need to be in the steering committee and you haven't been invited, please contact us to discuss that. Otherwise, the usual suspects tomorrow at 11. Is there any other announcements that people would like to make before we get started with the main business? Okay, I don't see anybody speaking up. So I'll briefly review our schedule for upcoming meetings and mini seminars. Next week, we have some industry representative talking about application of mechanistic model to design RTI prophylaxis. We have talks coming up by a review by Fred Adler of the viral evolution subgroup as an update, and Don Rice also, so that's more of a business meeting, updating us on the extensive work he's been doing on the dissemination outreach subgroup. May 20th, we have a single speaker, Rusty Irving, who is one of the developers of the original digital twin concept, who will be addressing us. May 27th, we have the noted science writer and former Nature editor, Philip Ball, speaking to us, along with another scientific speaker. And I should say that we're currently trying to line up additional speakers from the press. We have, thanks to Eric's work, connections to one of the main Associated Press reporters who cover science issues, and also one of the chief science writers for USA Today, who've agreed to speak to us. So we're looking forward to that. As always, we would appreciate your suggestions for future speakers. If you have connections, especially to clinical, experimental, or pharma or industry speakers, we definitely want this meeting to be connecting modelers to clinicians and experimentalists and to industry. And so your help in reminding potential speakers that they can be talking about clinical trials, they can be talking about drug discovery. You don't have to be a modeler to speak here. That would be much appreciated. As usual, our rules of the meeting, since the talks themselves are so short, we will hold questions until we get through both talks to make sure that our speakers have as much time as possible to give their presentations. If there's time for questions during the main meeting, we ask that people keep them general and short. And then we have our half-hour open pit discussion where people can take the gloves off and have whatever interactions they feel are appropriate. I will break in and give a 5-minute warning to our speakers. I hope you won't be offended, just to make sure that we stay on track so that both speakers get to finish their talks. Our first speaker today is Penelope Morel from Pittsburgh. As you know, I don't usually do an elaborate introduction. Better to save time for the talks themselves. And so I will turn it over to Penelope.
Okay, thank you, James. I guess I should share my screen. Okay, so a bit of a disclaimer before I start. I'm an immunologist. I'm not a modeler, and I have become involved, but as you'll see, I have become involved with several modeling groups over the last year. And so my perspective is really from someone who studies immunology and who is interested in this topic. And so I'm going to give you a perspective, not really my own work, but how I look at the problem and how I've been able to help with some modeling aspects. So this is kind of the overview of what I'm going to tell you about. Just, I'm sure I don't need to go into too much detail about SARS-CoV-2, the innate immune response, the adaptive immune response, and then some of the factors that distinguish mild and severe disease, and how two sort of modeling projects that I've been involved with. One is a sort of multi-scale tissue simulator using the PhysiCell approach, and another is modeling using a large model to develop a virtual patient cohort, which can be used to explore predisposition to severe disease. And these projects have been done mostly entirely in collaboration with Morgan Craig, Adrienne Jenner, Amber Smith, and Courtney Davis. And I'll try and if I have time, I'll end up with some new challenges and questions that I think are still remaining that would be of potential interest to pursue. So just a brief overview. We know SARS-CoV-2, it's an important pandemic right now. Over 150 million people have been infected with 3 million deaths, and obviously it's rampaging throughout countries such as India and Brazil. And interestingly, most individuals have asymptomatic or mild disease, but the problem is caused by a significant proportion, 15%, that require hospitalization, and 5% that develop severe disease. And this creates, these numbers may seem small, but when you have a large population of infected individuals, these numbers rapidly increase and become an issue for healthcare services and so forth. It's a complex disease, and the immune response is interesting to this virus. It's both important for viral elimination, but also it plays an important role in severe disease. And obviously, as you know, we now have several effective vaccines that have been developed, and these are beginning to have an impact in countries that are able to distribute this vaccine widely. Another issue, a couple of issues that are of concern still are that some individuals are developing some long-term health issues, so-called long haulers, and these may have relationship to the immune response to the virus, and obviously the emergence of new variants are posing a major challenge. So this is just overview of the symptoms and signs, which I think you're probably all familiar with. It's associated with pneumonia, loss of sense and smell, and it can be with severe disease. You can get multi-organ involvement and failure, Severe disease is associated with a number of comorbidities that are outlined here. Usually when we think of this disease, there's a person becomes infected, there's a short sort of prodromal period where before symptoms start, and this is a period where the disease can be transmitted from person to person. And following that symptoms or onset, there's a peak of viral load about 3 days, and then the immune system will kick in with antibody production. And if people go on to develop severe disease, this may occur around 10 days or so after the initial onset of symptoms. So this is sort of a general scheme of how we think about this. The virus is an RNA virus that enters cells through binding to specific receptors, the ACE2 receptor. When it enters the cell, it replicates its RNA to produce new viral particles that are released. During this process, cells will sense the presence of these viral proteins and RNA and will release innate immune signals such as type 1 interferon, as well as pro-inflammatory cytokines. And these play important roles both in preventing further disease, but also can contribute to the inflammatory process. And then we have production of specific T cells, antibodies, and B cells, which can neutralize and remove the virus. We do know that we have good adaptive T cell responses. This is one of the early studies that looked at CD4 T cell responses to a panel of epitopes that span the entire viral genome. And you can see that most of the T cells respond to proteins that are the structural proteins, the spike protein, the matrix protein, and the inner protein. But you do see responses to many of the open reading frames in the virus. Interestingly, these are all SARS recovered patients, and these are supposedly unexposed individuals. And we do see about 40% of people who have been apparently unexposed, or at least exposed perhaps to common cold coronaviruses, do have T cell and T cell responses that can recognize SARS-CoV-2 antigens. But in people who have recovered from disease, 100% of them have good CD4 responses as well as antibodies to the virus. So what do we know about factors that contribute to disease severity? Well, we know that I mentioned the interferon response is important, and if this response is blunted or delayed, this is likely to result in more severe disease. Severe disease is associated with a so-called cytokine storm with elevated levels of inflammatory cytokines. There is, a recruitment of neutrophils to the lung, which normally does not occur in viral infections. And also there's a depletion of the adaptive immune cells, such as CD4 and CD8 cells, which is associated with severe disease. So if we look at this in a sort of schematic, a normal mild infection, you'd have an early interferon or innate response that can prevent the spread of the virus. So the viral load peaks and then is cleared as the antibodies and T cells are elicited. In severe infection, this interferon or innate response is delayed initially and is less strong. So the virus can expand further, and then the immune response gets really out of control, and so you get an excessive innate immune response. So this is not only interferon, but also inflammatory cytokines such as IL-6. And the T cells, the adaptive response, really never take off in this situation. so you end up with more severe disease. And this is kind of schematically pictured again here, which I won't go through. The interferon, type 1 interferon, is not only important for stimulating the adaptive immune response, but it also prevents viral replication and protects additional cells from becoming further infected. And this is just another demonstration of how in a normal response you'll get good viral clearance with neutralizing antibodies, CD4 and CD8 T cells that can clear infected cells, whereas in a severe situation you have dysregulated cytokine production, excessive cytokine production, neutrophils coming in, and T cells are unable to clear the virus. So in terms of how we could think about modeling some of these things, these are some of the factors that are important, so-called viral factors, how viruses can interact with the interferon pathway to inhibit this pathway. They can also induce pro-inflammatory cytokines and induce sort of more inflammatory types of cell death, which leads to excess inflammation. There's a local tissue immune response, which is important. And then the lymph node response, which allows for the active generation of antibodies, CD4 and CD8 T cells. So I'm going to just show you some of the results that we have been working on with the Physicell model. So we've been focusing on the immune group with the sort of local tissue immune response to the virus. And this is sort of a general scheme of how this works. We have a tissue grid, which is representing the lung. And these are epithelial cells that can become infected with virus. And then we have various they can become infected and die. And then we have various immune cells, macrophages, neutrophils, dendritic cells, and T cells that sort of interact with the affected cell to either clear the virus or cause more damage. So I'm just going to show you, this is one of the latest representations of this, and you can see the blue cells here are the epithelial cells, and the virus is entering, from here, and the yellow, as they become infected, they turn yellow. And in this particular scenario, we're actually able to clear the virus through recruitment of macrophages, T cells, and some neutrophils. And if you look at these here, you can see the virus eventually gets cleared. We have activated macrophages coming and going down. And then I think over here you can also see the diffusion of various chemokines and the interferon pathway as this occurs over time. And so this scenario, this obviously we're modeling a good clearance of the virus, and I have to say that it's actually proven difficult to actually clear most of the early attempts with this. Either the virus or the immune system would just kill the entire tissue. But under this these conditions, we're able to clear the virus. And so I think this gives us a good opportunity then to modulate some of the factors, the immune factors and viral factors, and to determine how we may go from mild to more severe disease. So I'm just, in the sake of time, I'm really just giving you a very quick overview of what we've been doing here. But this other approach has been, this is Morgan Craig's group really pioneered this type of approach, and they generate a large mechanistic model of the immune response to SARS-CoV-2, and this involves virus, infected cells, macrophages, neutrophils, and T cells. And this is the large model is validated with a number of data sets, infection from many different sources. And using, once this has been, you can find details of all of this in this bioarchive preprint that was recently uploaded. But the interesting part of this is that using this model, we created a cohort of about 200 virtual patients. And by varying the range, plus or minus 20% of 7 parameters in the model. And these parameters were all related to macrophage, IL-6, and interferon production. And so each individual patient has a particular value for these parameters, and looking at the how these are put through the model, and then the values for each individual patient are it's assessed to make sure that they fall for most of these readouts, that they fall within known physiological ranges. So for viral load or for interferon production. And so then a patient can be picked based on that, and then is assigned to this cohort of 200 patients. And this, then we can look at the infection with SARS-CoV-2 and look at the outcome of the infection. And so just to sort of orient you here, this value is sort of a value that corresponds to high degree of, the higher this value is, the worse the disease is. So it's a combination of the IL-6 concentration, the number of neutrophils, and the actually not undamaged, but damaged lung tissue. And so what you can see in these graphs is that we have the 200 patients here that are all ordered in terms of inflammatory markers. So the ones up here with the highest have the highest inflammatory marker. So these are basically the 200 patients in order. And we plot these in relationship to various factors. And obviously IL-6 is included in this marker, so it's not surprising that you see IL-6 levels really follow quite closely this order. One thing I think that was of interest, and you can see also here, this is the T cell level, which as patients get more severe disease in this model, the T cells are reduced in number, which is what we see. One thing that was of interest here is, if you look, this is the time for the interferon peak. So this, I mentioned to you that delayed interferon is a bad prognosticator for disease. And here we see that if the peak of interferon is around 2 days, these patients cluster with a low level of inflammation marker. And if the peak is above 4 days, then this seems to sort of correspond to the patients with more severe disease.
I have to break in with my 5-minute warning.
Okay, I'm almost. And so this, we can then look to see what parameters are driving this factor, and there are 2 main ones that we can see here. So just again, these are the 200 patients with that are ordered by and then here is the value of the differentiation rate, one of the parameters that was used to distinguish the patients. And again, this is the red ones here have the low less than 2 of the interferon peak, and you can see that these, as this parameter increases, you see a change in the interferon peak level. This is also to some extent correlated with the production rate of interferon. what I just wanted to finish up with is just some new challenges and questions that remain. You know, how durable are memory responses post-infection, post-vaccination? What is long COVID, and how is the immune system involved? And what will the impact of new viral variants be on the pandemic? And we're already seeing some of that effect in India. And obviously the big questions are how effective are vaccines, and how can we improve vaccination strategies as well as distribution of vaccines. This is a recent study that showed that memory responses in patients that had mostly mild disease are quite persistent, and certainly at 1 and 6 months. It's suggested also that actually responses to vaccine are even more persistent than mild disease. So I think that's a good point. Just to orient you, these are all different types of immune responses, and the black here have antibodies as well as CD4 and CD8 T cells to they have all 5 of these responses. The one that seems to fall out most quickly is the CD8 T cell response. I won't really go into this, but this is long COVID, and there are a number of patients you may have seen who develop these long-lasting symptoms, and this is going to be an issue that we will have to address from the immune perspective in the future. And then, of course, the viral variants, as we know, could escape. They tend to improve the infectivity because they can bind to the receptor much better, or they cause more escape from the immune system, so that they escape may be more likely to lead to more severe disease. And so I think we're at risk of this type of situation, especially since countries such as Brazil and now India are unable to control their infections. And so this just becomes a sort of real breeding ground for new variants. And so this is going to be a problem that we will have to face. And so I just want to acknowledge really the modelers in all of this. I'm just really reporting on as I say, my role has really been to try and keep the immunology correct and up to speed. And but the modeling work that I've showed you has really been done by all of these people, with particularly Adrienne Jenner, who's done most of the legwork on all of this. And I will stop there.
Thank you very much. We really appreciate your willingness to speak in such a compressed format, but I think it's a wonderful presentation, a lot of interesting biology, and I hope we'll make some additional contacts and some new collaborations out of this. So thank you again. Now we'll move on. We'll hold the questions until the next talk is over.